Okay, so welcome to this next video in which we are discussing the acute inflammatory response and anti-inflammatory drugs. Now at the moment we're still discussing the acute inflammatory response and we're still actually on type 1 activation. Right, so we've discussed that if we can get myosin light chain here phosphorylated within our endothelial cell, then this will cause the contraction of the contractile units within the endothelial cell, which will cause the pulling apart of the tight junctions and the adherens junctions, which are between neighbouring endothelial cells here. And that will open up gaps between the endothelial cells, which will allow fluid from the blood and also proteins that are within the blood to leave the blood and go into the interstitial space of the of the infected tissue, basically. Okay, and this is going to be very powerful because all of the proteins that are within the blood can now go into the interstitial fluid, and many of these are going to play very important roles, which we'll see in a moment. Okay, but we've discussed that to get this phosphorylation of the myosin light chain, you don't just need to activate the myosin light chain kinase, which puts that phosphate group on. You also need to inactivate this myosin light chain phosphatase, which takes it off. There is no point in activating myosin light chain kinase to put the phosphate group on if you're not going to inactivate the enzyme which takes it off again. So we now need to look at what pathway within type 1 activation leads to myosin light chain phosphatase inactivation, and this is where we're going to come back to this beta gamma subunit that we have abandoned for so long. So remember, histamine stimulated the histamine receptor type 1 on the surface of the endothelial cell, which then catalyzed the conversion of it catalyzed the breaking off of this guanosine diphosphate and the binding on of the guanosine triphosphate. Okay, so you've got the alpha-Q GTP, which went off to cause the release of calcium, which has caused everything so far. It's caused the production of prostacyclin, the production of nitric oxide, and the activation of myosin light chain kinase. But now this beta-gamma subunit is going to have some stardom, okay, because it's going to come and it's going to be the part which results in the inactivation of myosin light chain phosphatase. So let's have a look at the pathway by which it inactivates myosin light chain phosphatase. Okay, so here is our beta-gamma subunit here. So basically, the beta-gamma subunit is going to activate something known as a Rho guanine nucleotide exchange factor. Okay, so let's call this structure here our Rho guanine nucleotide exchange factor. So, Rho guanine nucleotide exchange factor. Now, people have a acronym for guanine nucleotide exchange factor. So it's often just abbreviated to GEF for short, GEF. So this structure that the beta-gamma subunit over here is going to activate is often abbreviated just to a Rho, GEF. Okay, now basically this is going to activate a G protein. Okay, it's going to exchange the guanine nucleotide. It's going to take the guanosine diphosphate off of a G protein, and it's going to activate the G protein by putting guanosine triphosphate on. So what is this G protein it's going to act on? Well, it's this G protein Rho. Okay, now Rho is not a heterotrimeric G protein. It's not like GQ. It's a monomeric G protein, so it doesn't consist of 3 separate subunits. It only consists of 1. Okay, so here is Rho bound to GDP in its inactive state. When you activate this Rho guanine nucleotide exchange factor, it will catalyze the conversion of Rho with catalyze the conversion where you break off the GDP from the Rho and bind instead the guanosine triphosphate to this Rho protein. Now, what does Rho actually stand for? Well, basically, Rho, okay, so Rho here, this stands for Ras homology. Okay, so RAS is a huge, great family of monomeric G proteins. So this Rho G protein is very similar to the RAS G proteins. So RAS homology is what Rho stands for. The R comes from RAS, HO comes from homology. Okay, so the Rho GTP is then going to go on to activate the Rho-dependent kinase. Okay, so this is going to activate another enzyme known as Rho-dependent kinase. And this Rho-dependent kinase is then going to phosphorylate the myosin light chain phosphatase and inactivate it. Okay, so this is the Rho-dependent kinase. And I think we'll colour in the Rho-dependent kinase in purple. Okay, so this is the Rho-dependent kinase here. Okay, and it's now going to phosphorylate and inactivate myosin light chain phosphatase. Okay, so this is how you inactivate the myosin light chain phosphatase enzymes within the cytoplasm of the type 1 activated endothelial cell. So the beta-gamma subunit activates this Rho guanine nucleotide exchange factor, which then activates this G protein, Rho, which stands for RAS homology, by breaking the guanosine diphosphate that Rho has bound to it off, and binding a guanosine triphosphate instead onto that Rho G protein. The Rho G protein with guanosine triphosphate bound to it then binds to and activates this Rho-dependent kinase, which will add a phosphate group onto and inactivate the myosin light chain phosphatase. So this stick with a blunt end on like this means inhibits. So it's going to inhibit the myosin light chain phosphatase. So myosin light chain phosphatase is gone, basically. It's boomed out. It's inhibited. Okay, so now when the calcium calmodulin complexes activate the myosin light chain kinase, that's going to go around phosphorylating these myosin light chains on the actin filaments, and that will begin the process of contraction of these sarcomere-like structures within the endothelial cell, and that will cause these tight junctions and adherens junctions within the endothelial cell to be broken apart, basically. And this will open up a gap between these type 1 activated endothelial cells. Now this is especially important in the capillaries and post-capillary venules. And basically what's going to happen is that fluid from the bloodstream is going to be able to leave the bloodstream, and also with proteins from the bloodstream, they're going to be able to leave the bloodstream through these gaps now which exist between the endothelial cells, and they're going to be able to go into the interstitial space. Now, this new fluid that you are bringing into the interstitial space of this infected tissue is what's known as an inflammatory exudate. Okay, so this is a good keyword, so let me bring this up. So it's going to cause what's known as an inflammatory exudate, often just referred to as an exudate. Okay, now what is the purpose of this inflammatory exudate? Well, it's going to bring in a huge number of proteins from the blood which are going to help deal with this infection. Okay, so one thing that's going to be brought in is you're going to bring in complement proteins from the blood which are going to help attack the pathogen, and they produce 3 overall end results. So the complement proteins are a bunch of inactive proteins which are circulating within the bloodstream. Okay, now when they come into contact with the microbe, then they begin the complement cascades. Okay, now the overall results of the complement cascades, there are 3 overall results. One of them is that you end up coating the microbe in C3b proteins. So you bind C3b to glycoproteins all over the microbe, and so microbes end up coated in this complement protein known as C3b. Okay, so let me colour this in. So we'll colour in C3b in blue here. Okay, so all of these are C3b proteins which are now coating the surface of this microbe. Okay, now what does C3b do to the microbe? Why is it so effectively devastating for this microbe? Well, basically it's going to act as what's known as an opsonin. Okay, now this is skipping ahead a bit because we haven't actually got to the recruitment of leukocytes, the recruitment of white blood cells from the bloodstream yet. However, very soon we will recruit white blood cells from the bloodstream, and two of the major types of white blood cells we're going to end up with in the interstitial space are neutrophils and macrophages. Okay, now both neutrophils and macrophages are what are known as phagocytes. Okay, so these are both what are known as phagocytes, which means basically that they engulf pathogens and will digest them. So let me show you this on another piece of paper now. Okay, so the process of phagocytosis is what phagocytes undertake. Okay, so if this is our phagocyte here, then what's going to happen is it's going to firstly bind to the microbes. We'll show this is our little microbe here. And it has to bind to things on the surface of the microbe. Okay, so the phagocyte has receptors for ligands on the surface of the microbe. Now, one of the ligands that it has a receptor for is C3b. So on the surface of the phagocyte, there will be C3b receptors. So if your microbe is covered in C3b, it's basically a giveaway sign to the phagocytic cell that it should phagocytose this microbe, because usually the phagocyte has a bit of a debate about it, i.e. as to whether this cell really needs to be phagocytosed, because obviously it doesn't want to phagocytose healthy human cells. This could potentially be extremely dangerous if it were able to phagocytose any old cell. So it needs to be sure that the cell it's about to phagocytose is actually a microbe, and so it ums and ahs about it a bit. Now, if your cell is covered in C3b, then it's a giveaway sign that this cell is no good, basically, and should be phagocytosed. And that's what is meant by opsonization, okay, which is what C3b does. It increases the probability that a microbe will be phagocytosed. Now, the process of phagocytosis then, it firstly consists of this microbe binding to the surface of the phagocyte. Then what will happen is the membrane will invaginate around this microbe. So let's show this happening. So the membrane will invaginate around our microbe here. And then what will gradually happen is that you'll pinch this membrane off here. So I'll just highlight the microbe in again. So here's our microbe in red. You'll pinch this membrane off, and you'll get a separate little vesicle within the phagocytic cell. Okay, so here's our phagocyte again, and now we've got this vesicle inside with our microbe within it. Okay, so let me just colour that in again. So the microbe's in red here. Okay, now that vesicle, that special vesicle with the microbe contained within it, is what's known as a phagosome. Okay, and what will then happen is that you have other vesicle structures within the cytoplasm of this phagocyte, and these are known as lysosomes. Okay, so these are lysosomes, and they are full with enzymes known as lysozymes. Okay, also called lytic enzymes, which will break down the pathogen if they come into contact with it. So what's going to happen is you're going to fuse these lysosomes, which contain very deadly enzymes, with the phagosome and release these horrible enzymes all over the microbe that's within your phagosome. And that will destroy the microbe, basically, is the hope anyway. And this whole process of engulfing bacteria and then destroying them is known as phagocytosis. Okay, so that's one of the functions of the complement cascade, that it's going to increase phagocytosis. Now, we can't completely appreciate this yet because we don't know where the neutrophils and the macrophages come from, but trust me, they will come, okay? And when they are there, C3b is going to play a big part in helping them to actually phagocytose the right cells. Okay, the next function that complement plays is that it generates two molecules known as C3A and C5A, which are what are known as the Fiber Toxins, okay, because of their involvement in anaphylactic shock. Okay, so Fiber Toxins. Okay, so what do these molecules do? Well, when the complement cascades are established, you end up producing C3A and C5A, and basically they have receptors on mast cells. So remember mast cells, they were this sentinel cell that is dotted around all over your body. So you have it all over your body, in all of the tissues of your body, and they're filled with these granules which contain histamine. Okay, so let's put in the histamine as these turquoise dots here. Okay, so these are histamine granules within our mast cell. And basically, we remember that the mast cells degranulated. They released histamine into the interstitial fluid in response to detecting pathogen-associated molecular patterns with their pattern recognition receptors, and then that signified to them that there was a pathogen present. Okay, well now what we're going to get is a positive feedback loop, basically, because when you generate C3A and C5A, there are receptors for C3A and C5A on the surface of mast cells. So I'll just label this up as a mast cell. Okay, so let's let this here be a C5A receptor, and let's let the other one be a C3A receptor. So mast cells have receptors for both of the Fiber Toxins, C5A and C3A. So these Fiber Toxins will be released by the complement cascades, which are occurring on the surface of microbes, and they will diffuse over to the mast cells, and they will activate their respective receptors on mast cells, and they will cause more degranulation of mast cells. So they'll cause more release of histamine. So histamine release caused the type 1 activation of the endothelial cells, which is all of what we've studied so far. Type 1 activation of endothelial cells results in complement being recruited to the site of infection, and this complement is then producing these Fiber Toxins, which then positively feedbacks the whole loop and activates mast cells to release even more histamine. So this is a nice positive feedback loop. Okay, finally, the other thing that the complement cascade does, well, the complement cascades do, is that they produce membrane attack complexes. Now these are structures that are capable of attacking microbes in their own right, so they don't need macrophages to help them to actually kill the microbe. So basically, membrane attack complexes, or MACs for short, they are little tubes in the surface of the microbe. So if this is our microbe here, then you will produce this tube that goes through the membrane of the microbe, whether it's a virus with a membrane, whether it's a bacterial cell or a fungal cell or a protist cell. It's going to produce this tube that goes through the membrane of the cell, and this destroys the integrity of that membrane, basically. It destroys the ability of the membrane to control what can pass into the cytoplasm of the cell. So what starts happening is that water moves into the cell via osmosis, because the concentration of solute in the cytoplasm is much higher than the concentration of solute in the extracellular fluid. So water wants to move in. There is an osmotic gradient favouring the movement of water in. The reason water doesn't usually move in is that it struggles to cross the phospholipid bilayer, but now that we've stuck this tube in, water will move in, and that's going to swell this cell. The volume will go up and up and up until the cell swells and then bursts. And when you get a cell bursting because of movement of water into it via osmosis, it's what is known as osmotic lysis. Okay, so this is the way that complement cascades can directly destroy microbes, by making them burst into smithereens, basically. Okay, so that's the purpose of bringing the complement proteins into the site of infection, that they're going to coat microbes in C3b, which is going to lead to opsonization of those microbes. They're going to have this positive feedback loop where they create C3a, C5a, which are going to cause mast cell degranulation, and they're also going to form these membrane attack complexes, which will attack the microbes directly and hopefully lead to osmotic lysis of those microbes. Okay, now you bring other proteins in the inflammatory exudate. You also bring in the coagulation factors. So other proteins you bring in from the blood are coagulation factors. Now, coagulation factors usually have extremely boring lives. They go round and round in the bloodstream in this inert, inactive form, and they're never ever allowed to see certain molecules. They're never ever allowed to see collagen or tissue factor. Now, suddenly, we have opened up these gaps between endothelial cells in this site of infection, and the coagulation factors can fit through them, and they're suddenly into the interstitial space, somewhere they were never ever allowed before. Okay, now in the interstitial space, there is collagen and there is tissue factor. They were never exposed to these before. All they saw before was the other contents of the blood and the apical surface of the endothelial cells, neither of which have collagen or tissue factor. Okay, suddenly in the interstitial space there is collagen galore, or the extracellular matrix is made up of collagen, and all of the cells around have tissue factor on their surface, and these two molecules trigger the intrinsic and extrinsic coagulation cascades. So collagen triggers the intrinsic coagulation cascade, tissue factor triggers the extrinsic coagulation cascade, also known as the tissue factor pathway, and this leads overall, the big picture is that it leads to the conversion of a protein known as fibrinogen, or factor 1. Okay, it's going to lead to fibrinogen, which is also an old name for fibrinogen is factor 1. It is the coagulation factor 1. Being converted into fibrin, which is factor 1a, and I'm going to drop the factor now, I'm just going to write 1a. Okay, and then these factor 1a's are then assembled into huge great polymers known as fibrin strands. Okay, so you produce huge great polymers, huge strands, which form a very dense meshwork basically. So the interstitial space gets filled up with this huge great spider's web of fibrin strands. Okay, so that's what you should imagine it as, a really dense spider's web that you're forming. Okay, now what is the point of this? Well, the point of it is that you're going to produce a meshwork around all of these microbe cells, okay? And that's going to hopefully contain the microbe cell and stop it from moving elsewhere and spreading elsewhere, because the nightmare scenario is that it gets into the blood and then can disseminate all around the body. So if we build a cage of fibrin around it, then that hopefully will contain it where it is and stop it from spreading elsewhere. So that's the function of these coagulation factors being brought in. Okay, and before we just move on, I want to talk about one thing that I might have missed earlier on, which is that when you form this inflammatory exudate, where did we put this? Okay, when you form this inflammatory exudate, you're bringing in all this new fluid into this area of inflammation. Okay, now that's going to cause swelling of this area. Okay, now there's another great Latin word for swelling. Okay, so it's tumor, and this is another one of those pillars of inflammation. I just want to mention it because we're about to see the final two pillars of inflammation. So remember, there are 5 pillars of inflammation. We've had rubor and calor so far, redness and hotness, because of increased blood flow to the affected area. We've now had tumor because of increased vascular permeability. What we're about to see is the final two, which are dolor and functiolysi, which means pain is what dolor means, and functiolysi means loss of function. Okay, and those are going to be because of, well, not solely because of, but a major component of this is because of the kallikrein-kinin system. Okay, so the final important thing, majorly important thing, that's going to be activated by increased vascular permeability, i.e. that's going to come out of the blood and then is going to set off some sort of cascade, is what's known as the kallikrein-kinin system. So in the inflammatory exudate, we've seen complement proteins. We've now seen the coagulation factors. Okay, what we're now going to look at is the kallikrein-kinin system. So the complement proteins are often referred to as the complement cascade. The coagulation factors are often referred to as the coagulation cascade. The kallikrein-kinin system is often referred to as the kinin cascade. Okay, so what happens here? Well, certain proteins are going to come out of the blood, okay, so proteins that are made in the liver, and by the way, all of these are made in the liver. Coagulation factors, complement proteins, they're all made in the liver. Now, the precursors to the kallikrein-kinin system, there's only 3 of them really. There's Hageman factor, also known as coagulation factor 12, which is really important in setting off the intrinsic coagulation cascade. Then there's prekallikrein, also known as Fletcher factor, and finally there's high molecular weight kininogen. Okay, they all come out of the blood, and when they come out of the blood, they activate a cascade known as the kallikrein-kinin system, or the kinin cascade. And the overall result of this, and if you want to know the details of what happens, I have a whole video on the kallikrein-kinin system. The overall big picture is that you produce bradykinin, for one, okay, and this is produced from the high molecular weight kininogen, which comes in from the blood, and you also produce calladin, which is produced from the low molecular weight kininogen, which isn't actually within the blood, it's already in the interstitial fluid. All cells of the body produce low molecular weight kininogen. It's never usually activated to calladin because it requires the activation of the prekallikrein or the Fletcher factor that was in the blood, and that usually is circulating within the blood in an inactive form and is only activated when it comes out. Okay, so bradykinin and calladin, what do these now do? They're being produced because of increased vascular permeability, which has brought out these proteins from the blood into the interstitial fluid where they've been activated, and they're now producing bradykinin and calladin. What do these now do? Well, they go back to the endothelial cells. Okay, so they're going to diffuse back to the endothelial cells, and they're also going to lead to a positive feedback mechanism, because these endothelial cells have a G protein coupled receptor on them, which is also GQ coupled, okay, so there's the 7 transmembrane domains, and will also trigger type 1 activation if it's activated, and this receptor is susceptible to bradykinin and calladin, and for this reason it's referred to as the bradykinin type 2 receptor, or the B2 receptor. So this is the bradykinin 2 receptor. So both bradykinin and calladin, which is almost an identical molecule, it varies by a single lysine amino acid, okay, both of these will bind to this bradykinin 2 receptor that is on the surface of the endothelial cells, and they will trigger type 1 activation. So they're going to trigger a positive feedback loop. So you're producing these because the endothelial cells underwent this change where vascular permeability was increased, and now they're going to go back and trigger more type 1 activation, which will result in more increase in vascular permeability. Okay, so the other thing these two molecules do, apart from going and activating the B2 receptors on the endothelial cells, is that they also activate pain neurons in the local area. So they're going to go and trigger action potentials in the pain neurons, and these will go up to the brain and trigger pain, and they'll also trigger loss of function. So you'll stop using the affected tissue. Okay, so these are going to lead to the final 2 pillars of inflammation: dolor, pain, and functio lysae, which means loss of function. Okay, right. So, those are the final things that bradykinin and calladin do. And bradykinin is often called the most powerful pain-inducing chemical known to man. Its ability to cause pain is second to no molecule that we know. It is the most capable molecule of causing pain, basically. Okay, so that's the purpose of increasing vascular permeability, that you get the complement cascades, the coagulation cascades, and the kinin cascades starting as soon as you bring their precursor proteins into the interstitial fluid. Okay, right, so let's now turn our attention to the final thing which happens in type 1 activated endothelial cells, which is that you're going to start recruiting neutrophils. Okay, so we'll turn our attention to that in the next video.